Let's start. Okay, so hello everyone. Welcome to the live demonstration of advanced microscopy. So here we will explain about the confocal microscopy, which is equipped with the super resolution module, that is NSPARC. So first we will explain the components of this microscope. So this one is your computer and monitor, which will control the software of this microscope. And here, this one is your LED box, and this one is your camera. This one is your main microscope, and here we have joystick, which will control the stage of your microscope. And along with this, we have these two components. This one is your LED box controller, and here we have chamber. If you want to do live cell imaging, then it will control your CO2, your temperature, and humidity. So the controller of this chamber is placed here, and your microscope controller is this. And then we have next component. This box is for confocal scanner, and the black box is for this one is Nikon and Spark, and this one is for your super resolution controller. Along with this, we have this laser controller and power controller box X controller, and then this one is your laser box. We have six different laser systems: 405, 455, 488, 514, 561, and 640. And this one is for switching on these lasers. So now we will use 450 laser, 488, 561, because the slide we are going to use is stained with DAPI, and we have visualized mitochondria with mitotracker green, which will be visualized with 488. And then we have stained actin filaments with rhodamine phalloidin, which will be visualized with this 561 laser system, that is TRITC. And this box is for turf laser system. And this one is your detector, which is equipped with PMT detector. So next, we'll start with software. So this Nikon confocal microscopy is controlled by this NIS element, and this software has different functions. So here are 7 components. First one is live. So if you want to see your sample or your slide, which is having cells, mammalian cells, bacterial cells, yeast cells, or any other cells, you will do live, and there you can see your cells and components on the screen. For capturing, you will use this capture option, and if you want to find your samples, you can click here, and the software will find automatically your samples or cells. For autofocus, you have to click this, and this software and this microscope is having one advanced feature, that is auto signal AI. So here you don't need to do anything. It will focus your sample, it will control your signals, and it will find the place of your sample, and it will give you a best image. Then this one is ND acquire system. So if you want to get 3D image of your samples or Z stacking, Z stacking, you need to use this option. And then there is one feature, that is large image. So this one is for image staging. So for like this one is your 5 by 5 visualized area. If you want to capture large images, like few micrometers here or few micrometers right or above, then you will use this large image option. And then in this software, there are main these components: dia, AX and Spark, API, fluorescence, then dia and API. So if you want to visualize your samples by using microscope and your eyepiece, okay, then you will click this. But if you want to visualize your sample in confocal mode or fluorescence microscopy on the screen like this, here, you have to click this option. And the another one is X in Spark. This is for super resolution imaging. And then if you want to visualize uh, your samples in fluorescence, standard fluorescence microscopy mode, then you will use this API and fluorescence microscopy mode on your screen. And then there are different, there is the mode for different channel selections. So here we are going to use DAPI. We have selected this FITC and TRITC. And we have not selected, this one is not yellow, that means we are not going to use this channel. So this option is for your channel setup. Apart from this, you can also add your channels by using these options, so there is no need of these. Again, here these are the components for your auto signal AI. You have to click this. Here there are several options: live, medium, fixed. So mainly we use this option. And this one is for auto laser off or shutter off, because you cannot visualize your sample continuous. It will lead to the sample bleaching. And then we have one denoise.ai option. So if you see that your sample is having so much noise, here you can see there are several noises. So this function will remove all these noises. So you have to click this. And again, we have two scanners. First one is resonant and the second one is galvano. So conventional microscope has these galvano scanner. But here in this microscope, since it is advanced version, it has resonant scanner. It will increase the scanning speed of your microscope. So for high scanning purpose, you need to use this scanner, but now we'll use Galvano. And here the other function is averaging for your speed.
How
Or
many times the same
Yours-
feed will be captured to increase the resolution?
and then again 12 times.
For how long it will expose?
Yes. Okay, so here are the options for your lasers which have been selected here. So from here, you can increase the intensity of your lasers. So for DAPI, you can increase here to here. So you you should see or check that your laser power or intensity should not be very high. And here you can use offset to this one is another function to minimize your noise, that is offset. Again, since this one is confocal microscopy, and I believe that you might have learned about the differences between standard fluorescence microscopy and confocal microscopy. So confocal microscopy has one pinhole which narrows the light path so that you can visualize a particular or very small area of your sample, and your sample cannot be bleached at the same time, and it will improve your resolution. But what happened in fluorescence microscopy, it captures the light from all over the area, and it limited with the resolution, and also your samples will be bleached out. And next, here these options are for your visualizing area or scan area. So based on the pixels, 2K, you can choose 2K by 2K, but this will give slow scanning, then 1 by 1K pixel, and 5
512 by
512
512.
512 by 512 pixel, and this will provide better scanning. So here we have choose this option. So next, here are the option for your objective. So this microscope is equipped with 4x, 10x, 20x, 40x, 60x, and 100x also. So now we have used 60x objective oil immersion for the visualization. Okay. And next, what we have?
Filter turret.
So from here, you can see the turret position of your microscope at the stage. Here you can see there are different objectives. So you can monitor from here that which objective is there at the turret position. Okay. Next, we'll start visualizing the samples in fluorescence mode, confocal mode, and super resolution. So we will demonstrate one by one to you. Okay, then we'll switch off the lights. First, you can show Krishna, we have kept the samples here. Just come to the stage. Here, your sample is placed. Okay, these are the mammalian cells. We have uh, fine. First, we'll do live. From here, as I mentioned, from here you can do live. Okay. So we'll change the area by using joystick. Here you can see. We'll find the best area to capture the images. So right now, we are finding the cells in confocal mode, but first we'll capture it in standard fluorescence microscopy mode. Okay. So from here, you can, this one is joystick. From here you can change your slide position, stays position in XY direction, and from here you can change the Z position. So the sample is very bright. You can see the red one is your mitochondria. It's too bright. It's too bright. Okay. Mitochondria is sustained with.
We can do what is about it.
We'll do first. We'll do live, and we will minimize the intensity. We have to increase clear and TCs. Okay, so the mitochondria is stained with, I think this one is lyso, a mitotracker red, and the actin filaments are in green channel. Okay, we will increase this green. You can see the changes on the screen. We'll decrease the TRITC. Okay, from here we will change. One more thing. Now it's fine. One more. fluorescence mode. So first we'll start capturing in fluorescence mode. Okay. Standard fluorescence microscopy. For this, we have to click this option. Okay. Then we'll do live. For this, we have to select channels. Oh, just wait. There is. I'm going to close this one. This one. So here you can see there are several noises. We can use this AI function. That is also auto signal AI. We'll give command to this option, this feature, and then it will do everything. So here we'll click this. You can see it has started its job. You can see the differences. It's changing the signal intensity. Okay. So this one is created by Auto Signal AI. And if, okay, so if we want to remove the noises, we'll do like one more. Okay. Then to reduce the noise, we'll click denoise.ai. Here. Then live. You can see it has reduced the noise. So here we'll change the position one more time. We are changing the Z option so that we can get better image. Yeah, I think this one is best. So now I'll change XY position. Okay, okay. This one also. Right. Find the composition.
Okay.
So now we have found a good area to capture the images or scan it. So here we'll do auto signal AI. It will do its job. Okay. So we have clicked it. Here we are about to find the best image. So it's changing the channels one by one and their intensity. First, uh, first it changes the green channel, then red, and then your dark. So here we will capture this image. This one is your confocal image. Okay, we'll keep it here so that we can compare it. Okay, then one image we will take with denoise.ai. You can see the noise has been reduced. So again, we'll capture this and here we'll keep it to compare. Then we'll go to X Inspa. So as I mentioned that this option is for super resolution imaging. Okay, so we'll click this. And so this module function, when you reduce your FOV to the pixel size of 107. So from here, we will change 107. Okay, so next we'll take one area where we can visualize it properly. Okay, then after this, we'll go to the deconvolution option.
Later. We can do deconvolution later. First, we can click one picture in NSPA.
NSPA.
Then we will
Fine.
compare it to
Okay.
deconvolution.
Fine. So we'll go to the confocal mode first so that we can take one picture of that area. We do live and then from here I will minimize this FOV like that. Here we will take super resolved images. So now we are taking in confocal mode for comparison. 107. We'll do live in confocal mode. You can see. This one is your confocal image of that zoomed FOV. Okay. Then we will capture it. We will stop live. Then we will capture it. Then it is captured. And then we will keep it for comparison. Okay. Again, we'll go to super resolution mode, which is X and Spark. Then we'll come here, and here you can see the same area has selected. And then we'll do
Capture.
not capture, deconvolution.
First we capture, then we'll deconvolute.
This we can do. So this is live image of that particular area. So here you can see this magenta or blue is your nucleus. These green filament structures are your mitochondria, and red, uh, this red color visualized area is your actin filaments. Okay. Then for to super resolve this image, we'll choose this option that is it's not deconvolution option. We'll click this. We'll go to the 2D deconvolution, and then it is same only. Okay. Then here are several options. You have to click it here. The deconvolution method will be blind. Mode will be this, and pinhole size 1. And magnification, we are taking it at 60x, this objective. And all the information you have to set, and then you have to deconvolute. Okay. So here it has started. It will take some time. So now here you can see your super resolved images. So from here, we will minimize this window. Okay. So from here, we can compare that this one is the image taken from the confocal microscopy without any noise reduction. Okay. And this one is, so here you can see there are several noise, some green noises are there, but in this image, there is no such noise. So this one is denoised by this feature. Yeah, okay. From this option, this denoise.ai. Okay. So again, this one is captured. This is the zoomed area. So while we zoom the your visualized area, your image get some blurry. So here you can see this is somewhat blurred, and it was captured in confocal microscopy. Then we changed it to the NSPARC super resolution mode. and this one is it's just we did live and captured, and after implementing the super resolution mode, here you can see super resolved images. So the structure and filaments and edges of mitochondrial filaments are well defined. You can see even the actin filaments are visualized, well defined as compared to this and this. Okay. You can just differentiate it by seeing the mitochondrial filaments. Okay. These are the super resolved images. So next, we will do the 3D imaging of 3D imaging or Z stacking of these cells. So
We'll close these windows and go live. Then we'll show you how we take Z stack from this NIS element.
okay. So as I said, first we'll do live. First we will maximize the F probability, maximum probability. We'll do live. Should I change the area because it has bleached now? Okay. This one is better. Change this.
This one's good.
This okay. Okay. So for 3D imaging or Z stacking, we'll go to this option as I mentioned earlier, that is NDF quad. Okay. So this window has several options. From here, you can do 3D imaging, 4D, 5D, and 6D. So if you have idea about these imaging systems, for this, it has several components. For time, it has option. For different position, it has option. For different channel, it has option. And for different stats, you have to click this uh,
Z option.
Z option. Okay. So for 3D imaging, we'll perform 3D imaging only right now. So we'll click this. And apart from this, there are three options, three module you can say. So if you select this, you have to decide which one be your top position and which one will be your bottom position. And by selecting this option, it will give you automatic a system from your middle point to the 3 micrometer above and 3 micrometer lower side. And from this option, you can choose your stacks in different ratios. Like from the middle point, you can choose 3 micrometer above and 1 micrometer bottom. Okay, so here we will choose this, and then we need to select. Now we need to select top and bottom. So first we'll go live. Okay, then we will reset the Z position.
So we take it at the
So now the Z position is above to your main sample position. So here we will select top, and then we will come back. Okay, so your image is again blurred, and here we will select bottom position and then run now. Okay. So it has started taking images at your different steps, and here is another option. It will take 4 minutes.
Because we have selected
Whatever.
Yeah.
Okay, we will avoid because it is taking so much time. Okay,
like this.
so here we'll keep it at 40. Okay. 40, then we'll start it again. Okay, again it's taking. We'll keep it 40. Okay, fine.
So currently it is capturing Z stacks in the confocal mode. So this window shows you the time calculated. It will take around 3.5 minutes to take this one 40 Z planes for this sample. So here in this window, uh, here window you can see it tells after how much distance it will take the distance of the Z steps it will capture. So this 0.145 is calculated through the Nyquist calculation, which we have read about in the course. So depending upon the Nyquist calculation, it tells us that how much the step size should be. If the step size is more than that or less than that, it will end up doing undersampling and oversampling. That's it. So this window tells us that these green steps it has taken, and this orange step it is taking right now, and these many steps remains to be captured. And this tells us that it has captured 33 steps out of 40. If you click the finish option, it will stop taking the Z stack, but will store the data till what it has taken. But if you abort it, then it will delete whole data that it has captured.
Pause it. Okay, so 11 seconds are remaining. So it's done now. Okay. So from here, so from here you can see.
By moving on this blue steps, you can you can move from one step to another.
And if you want to see it in this,
It might be this one.
so
Next one.
this one is
Next one.
MI.
So this one. Next one. This one.
Okay, so this one is your MIP image of this Z stack.
So if you can see by clicking this icon, it gives you the maximum intensity projection of this time lapse movie of the Z stack that we have taken. So if we maximum intensity projection, we have read about it in the course. So if we click it, show maximum intensity projection. This one, this one is the so this is the uh, I guess this is the this one is MIP and this one is the Z stack image that we have captured.
Okay, then next we'll this. Okay. So here, if you want to see your images in different directions or your Z stacks, from here, check this. Okay. In this direction, you can rotate the stacks, and then if you want to make a movie of these images or rotation of these stacks, you have to choose all these options. So here are several boxes. Here you will choose your image first, and then from here we'll choose rotations. We just need to drag it here. This one, this one. We'll select all these rotation modes. Okay, and now just you need to play it. So this one is best for if you want to check any internalization of any sample inside the cells. So this mode is best for this purpose. Okay. So after this, we will visualize some images in large mode, or we'll do image stitching. For this, first I close all these things. And we'll do live. Right now we are in right now we are in confocal mode. First we need to visualize your cells. This one is
No, no,
not this. Right, close this one also. Okay. So we have visualized the cells again. Okay. We'll freeze it. Then we'll go to large image. Okay. So it was captured previously. We have to we have to reset it. It has several options. From here, you have to select your channels, in which channel your sample will be visualized. You have to do here tick mark, and here you have to select whether you want to save your file or not. And okay, so from here, you have to select your area for large image capturing or image stitching. So first we'll go live. Okay, here those samples are there. And then we'll go up. Then, okay, so to start this, first we have to go at this option. We have to select this symbol. Shoot. This one. Okay, we'll select this, and then we'll go into the upward direction. Here you can see images are moving. Okay, so at the particular area, you have to select. From here, we will take images at this area. For this, we have to click it here. Then we'll go to the left direction. Okay. From here, it will take capturing. We'll go to downward. Then we'll click to this option, and then go towards the right direction. Then we will click it. Yeah. So we have selected the areas and then we'll run it. And you can see there's started stitching the images. Still, this one is confocal mode. So laser, lasers are changing. Laser channels. It will come. Now you can see we did live at this area, like this area, right? And after image stitching, you can see so many cells. So it has taken images at different positions in upward, downward, left and right, and then it stitches all those images to make a large image. Okay, so I think it's done for today. And if you have any question, you can ask or you can write in comment section, chat section. Okay, so the question is why did the mitochondria fluorescence color change in the high resolution? So here you can see if we go to the X confocal mode, the DAPI, FITC, TRITC means your nucleus and mitochondria and uh, that actin filament. These are in uh, blue, green, and red channels. So if you want to change your color of your channel, you can change it from here. Okay, from here you can change. So in this confocal mode, your mitochondria means this green color. It is in green, but in X in spark mode, it was selected in red color. Okay, only the color of your channel is changed. Baki, otherwise,
Otherwise, the cell part, cell organelle is same. Just the color assigned to that channel is different.
yes, your dye or fluorescent marker is same. Just the color of channel is changed. Okay. Any other question? If you have any question, you have asked later. You can ask later.
The sheet. Yeah, sure. The sheet that they can ask questions.
Okay. Sure.
Okay, so we are done for today. If someone has any question, they can update it in the sheet. Thank you all for joining.
Ma'am,